Hello and Assalamualaikum. So today I will present about the expression of bacterial genes, transgenic tobacco. So before that, I will introduce myself. My name is Muhammad Mirza bin Malik. I am from class M380-204E. So I will start with the introduction. Tobacco is widely used as a model plant system in transgenic research for several reasons. Its molecular genetic is well understood. Its genomic mapping is almost complete. Genetic transformation can be readily achieved. Tobacco plants survive well in vitro and under greenhouse conditions, and tobacco produce large amount of biomass. Tobacco is also regarded as one of the best systems for chloroplast transformation, which further enhance the effectiveness of tobacco for the expression of recombinant proteins. Tobacco plants can be used as living factories to produce proteins and enzymes, which can be extracted, purified, and used for the manufacturing of pharmaceuticals and other valuable industrial compounds such as biopolymers. One of the most serious problems in agriculture is the loss of productivity caused by plant pathogens. To cope with the constant rate of invasion by pathogens, plants evolve over time different mechanisms to protect themselves. For a plant to establish a successful self-defense response against pathogen, specific plant receptors need to promptly recognize the pathogenic elicitor molecule. So I will next continue to the gene used to produce the transgenic crop. So first we have Halobacterium halobium bacteriopsin. Gene under control the wound-inducible promoter used to develop transgenic tobacco plants resistant to Pseudomonas syringae, Tawachi via agrobacterium-mediated transformation. Bacteriopsin activates the self-defense mechanism in plants by enhancing proton pumping across the cell membrane. Second, the POP-A gene from the phytopathogenic bacterium Ralstonia solanacearum used to develop transgenic tobacco plant resistant to the violent fungus Phytophthora parasitica, VAR nicotiana. The elicitor protein POP-A from R. solanacearum has been shown to be an effective inducer of HRs in tobacco plants. Third, tobacco plants carrying the E. carotovora gene ESPL, which is responsible for OHL biosynthesis. Next, BT-Cry2AA gene transgenic plants expressing insecticidal crystal protein genes, such as Cry-A, Cry-A1A, Cry-A2A, and etc. From BT are known as BT transgenic plants. More commercial BT transgenic crop carry a single cry gene inserted into the chromosome that express the BT protein constitutively and usually at low levels. Next, the COM gene, DCHO-M gene, isolated from Actinomyces A19-49, encodes the enzyme cholesterol oxidase, CHOM, which has powerful insecticidal properties against the boll weevil larva. This enzyme acts by oxidizing cholesterol in the insect midgut epithelial membrane, disrupting the physical and functional properties of the membrane and causing death. We'll next move to the method used for gene transfer. So first we have agrobacterium-mediated transformation. The use of agrobacterium-mediated gene transfer as a method for introducing genes into tobacco plants. This method involves the use of the soil bacterium agrobacterium tumefaciens to transfer genes of interest into the plant genome. An example of this method is the transfer of the bar gene into tobacco plants to confer resistance to the herbicide Biolopos. Second, biolistic or particle bombardment. This method involves coating DNA onto microscopic particles and then using a gene gun to shoot the particles into plant cells, allowing DNA to be incorporated into the plant genome. An example of this method is the transfer of genes encoding insecticidal proteins from Bacillus thuringiensis (BT) into plants to confer resistance to insect pests. Third is plastic transformation. Method is to introduce foreign genes into the plastic genome of plants. This technique involves the insertion of genes into the plastic DNA, which is inherited maternally and has its own gene expression machinery. Plastic transformation offers several advantages, including high-level expression of foreign genes and the ability to express multiple genes from a single transformation event. One example of plastic transformation is the introduction of the tetanus toxin protein TTC into tobacco chloroplast. This was achieved by cloning the TTC gene into a chloroplast vector and transforming tobacco plants. So, we will next move to the workflow of gene transfer. First, the gene of interest is isolated and prepared for transfer. Next, then the gene is introduced into the plant cells method such as agrobacterial mediated transformation, plastic transformation, or biolystic method. Once the gene is successfully integrated into the plant genome, the transgenic plant are selected and screened for the expression of the introduced gene. And lastly, finally, the transgenic plants expressing the desired gene are further analyzed and evaluated for the production of recombinant protein or enzymes. We will move to the result of the application. So first, enhanced resistance. Plastic transformation has been used to express bacterial genes in tobacco, resulting in enhanced resistance to insect pests. For example, transgenic tobacco leaf expressing a Bt protein resulted in 100% mortality when fed to Cry1A resistant tobacco butworm, cotton bollworm, and big armyworm insects. Additionally, the expression of the bacterial 2,4-D monooxygenase gene lead to the development of herbicide-resistant transgenic crop plant. Second, pathogenic resistance. The Halobacterium halobium bacteriopsin gene BO expressed in transgenic tobacco resulted in enhanced resistance to Pseudomonas syringae. The AGR569 gene expression in transgenic tobacco confer resistance to Pseudomonas syringae. The carotovora gene ESPL, when introduced into tobacco plants, led to resistance against soft rot disease. Third is insect resistant. Transgenic tobacco expressing the Cry2AA2 gene exhibited high level of resistance to tobacco butworm, cotton bollworm, and big armyworm insects. Fourth, herbicide tolerance. The bacterial 2,4-D monooxygenase gene expression in transgenic tobacco resulted in the development of herbicide-resistant plants. Fifth, abiotic stress tolerance. Transgenic tobacco expressing the bacterial acylipid desaturase gene exhibited increased resistance to cold temperature. The introduction of the E. coli BAT-A and BAT-B genes into tobacco plants via agrobacterial mediated transformation led to the production of betaine, enabling the transgenic tobacco to grow in the presence of high salt concentration. 6. Phytoremediation. Transgenic tobacco plants expressing the E-KE-ONR gene show tolerance to and degradation of nitroaromatic and nitrate ester explosive. 7. Vaccines production. Transgenic tobacco expressing the TETC gene produce significant anti-TETC antibody response, demonstrating the potential for vaccine production. The expression of the intimin gene in transgenic tobacco induced an immune response in mice. So to conclude, recent discovery in genetics and proteomics have uncovered crucial genes in both bacteria and plants, including tobacco. These genes play pivotal roles in helping plants like tobacco withstand harsh conditions, fend off disease, pests, and toxins, and even produce beneficial substances such as pharmaceuticals. Understanding these genes in tobacco and other plants can significantly advance agricultural practice and industrial process. By manipulating the metabolic pathway in tobacco plants, we can optimize protein production and enhance crop quality. However, extracting these proteins from tobacco plants remains a challenging and costly task. Researchers are exploring alternative methods such as using tobacco cell cultures to simplify and reduce the cost of protein extraction. Despite the challenge, the potential benefits of harnessing tobacco's genetic capabilities for protein production are substantial, offering promising prospects for improving agriculture and industry. So this is the references from Jib S. Botakadedi, expression of bacterial genes in transgenic tobacco, method application and future prospect. Electron J. Biotechnol, 2007, July 15. So that's all from me. Thank you.